Hi, hello everyone. Welcome to Blumberr podcast. Cancer treatment. It's no longer just about chemotherapy, radiation, or surgery. Today, cancer care is becoming increasingly personalized. Scientists can now study the genetic blueprint of a tumor, detect cancer through a simple blood sample, monitor treatment responses in real time, and even develop therapies tailored to individual patients. Few researchers have been at the forefront of this transformation. And one of them is as closely as Dr. Prashant Kumar, who we have with us today. A cancer biologist, transitional researcher, and a precision oncology leader. Dr. Kumar has spent nearly 2 decades working across some of the world's leading scientific institutions, from the Max Planck Institute in Germany to UMASS Medical School in United States, A-Star Singapore, and now leading cutting-edge oncology innovations in India. His work spans genomics, liquid biopsy, biomarker discovery, precision medicine, artificial intelligence in healthcare, and therapeutic cancer vaccines. Today, we'll be exploring not just science, but also the person who is behind all of these innovations. Hello, Dr. Prashant Agarwal. Welcome on board. How are you doing today?
Very good. Thank you so much, Pushpa, for this invitation. And I'm looking forward to have a very prolific discussion and also touch base about the scientific journey, what I have been gone through and how the world is transforming, especially in the oncology space. So thank you again.
Thank you so much for joining us today. Doctor, you have told me once that it was the cloning of Dolly sheep and that article, that information, those were one of the moments when you decided that you would want to be a scientist. It was kind of a breakthrough into your fascination as a young child. What was that like? And when was that?
Yes, it absolutely. I think it was year 1996, and 1996 when Dolly came, and 1997 when Polly came, and we were growing up, and for us it was very fascinating.
Correct.
Because I truly believe that Dolly ship it's a rare scientific moment that profoundly shaped the imagination, and for us we imagined that something could be cloned. And from the adult cells, and what we learned that time, that from the adult cells things will be irreversible, and still retain the complete genetic blueprint and to create entire organism.
Correct.
So I truly believe that Dolly taught me, and my science is not about the just only accumulating the knowledge, but expanding the knowledge. And I think this was the time we were growing up, we were reading the news, and everywhere in the newspaper and the news channel, we were seeing the videos, and that actually fascinated me, that the research has no boundary. You cannot make your mind very rigid that, okay, this cannot be done, or this is something, a thumb rule. When you start expanding it, you start learning more, and I think we should all challenge the existing concept, because that is the only way you will grow and learn more.
True, very true. So, during your journey as a scientist, when you picked up science as your subject, when did you stop, the moment when science stopped being a subject and becoming a calling for you?
So see, all of us, when we grow, we are very naive, I would say. And doesn't matter it is science or you take business or commerce or arts. And I start now feeling, and I keep telling that, when I meet young kids, I tell that this book now I can read in 1 hour, which takes in class 7th and 8th a year to prepare.
Yeah.
And why this happened? Because we learned over the time, we grow over the time, our concept changes. And I truly believe that this is something I was saying, that witness of the cloning of Dolly and sheep for people like me or a young student, it was really very exciting. And I truly understand that biology is not static. Believe me, the discovery more from lab to clinic that influenced the patient outcomes. And for me, every time when we were growing up and my journey, I start meeting the people, leaders across the world, lot of discoveries, and that changed my perspective that no, it has not a single dimension, but it had multiple dimensions, and we should actually open to think more, grow more, and learn more.
Very true. As much as we gain knowledge, that is when we internally evolve and we would be able to contribute also. Doctor, you have chosen oncology as your chosen career beyond biology. Why was that? And what created that enthusiasm or interest in oncology?
So this is often say I didn't choose cancer
Hmm.
because it was easy.
Hmm.
It was not never easy to change or choose a cancer biology.
Hmm. Hmm.
I was drawn to it
Okay.
because it was fascinating and very challenging.
Hmm.
And cancer, after I would say, what is something what fascinates cancer is it virtually touch every family and every community.
Hmm.
And this is where I truly believe that the oncology was gone through a revolution, and there is a human purpose. So this is where I started believing that when I think I was first introduced when I was in Germany, Max Planck. I started working on gastric cancer, and that time I was much more in the lab. But when I realized that, and I was questioning myself always that I am working in a small bench, or I used to tell Max Planck a matchbox. We used to go early morning and leaving in the evening, and we all are working. I never thought that me doing some work would make an impact. And trust me, if all of us will understand that you are just doing a pipetting, you may feel that okay, we are coming and doing routinely a pipetting, and does it merely going to make an impact? But then I realized that no, the discovery we made in the lab, which we feel that okay, we are just coming and doing routine work, or we are changing or postdoctoral fellowship, but it has a much bigger meaning, and the bigger meaning is to discovery, the innovation. And this is where my journey about the cancer started. And the more and more I went deeper and learned the very bigger part and the serious part, that the discovery is going to make an impact for the cancer patient treatment decisions. That actually is my whole perspective to deal with the oncology.
I mean, it seems that you had a lot of curiosity and were driven by purpose when you had chosen oncology in Germany in the Max facility, right? So, as you said, it is a cubicle and you still are sitting in the lab. So, that curiosity or purpose, has it been a big driver in your career on the whole?
Yes, that's an amazing question. And I think the curiosity ignites the journey. Trust me. And again, depending on which field, which area you choose, it fuels you. And I do remember the countless hours in the lab. And especially, I remember during my PhD time in Germany, doesn't matter it was Diwali or Holi, because eventually we don't have Diwali and Holi in Germany.
Yeah.
So for me, only Diwali, Holi, or any Indian festival was on the phone. When you call your parents and say Happy Diwali or Happy Holi. Christmas maybe, but even though we coming from India, people expect that okay, you are not celebrating Christmas in length.
Yeah.
We are coming in the lab. So it's the countless hours spending in the lab. But believe me, scientists are by nature driven by questions. Whenever you think about scientists and think about the scientists, and for me, the question start arising pretty early stage of my career, that why does cancer arise? Or why some of the patient, they respond to the treatment, or why and how can we detect cancer early? So I would say that curiosity is what keep us pushing boundaries. But what is also important is the purpose. Trust me, purpose actually make you think and deeper, that how can you endure your force, and how you can discovery, because the discovery can impact and the treatment, decision, diagnosis. But I think the cancer driven drives the discovery, but purpose drives the direction. So this is very important, discovery and direction. And this is where I truly believe that any of us who are driven by the curiosity, but what is also important at some point of the time, that what is the direction and which direction we are bringing our curiosity to succeed.
Understood. Wow, yeah. So, doctor, when you studied in IISc, one of the premier colleges in India, and then you moved to Max Planck Institute in Germany. It is one of the another premier research institutes across the world. So, what was the cultural shock you had when you moved from here to there? Be it scientific, be it personal, how was it?
Yeah, so again, this is one of the very incredible question you're asking. And remember, 15, 20 years back when you have been trained in India and especially in Indian Institute of Science, which I truly believe that that was the foundation for me to get exposed to the world class science, and truly blessed to have in Indian Institute of Science spent 18 months before I took a decision that I'll move to Max Planck, Germany, for my PhD.
Okay.
So I would say it was curiosity-driven research. What I found when I going to any part of the world, another part of the world, what is very important is challenging the ideas. What I feel very different when I moved to abroad, and disagreement, which should not be disrespected. When you go, it doesn't matter you are a very young PhD student or a postdoc. You can always make a disagreement for a research based questions or research discussion. It doesn't matter which who is sitting other side of the table. If a Nobel laureate is sitting, he would have all patience to listen a young student that how I think differently. So I think that is something I felt when I moved from India to abroad. I felt that the comfort zone where I can question
Question.
even most established concept. If there's most established concept, I can argue that I may agree, I may not agree. And I think science has been driven over the time by the data. I always call that this is the time, it is a data driven science. It is not a hypothesis driven science. Now, if I will make any hypothesis, you will question me that show me the data for me to prove my hypothesis. So I would say that it is actually made me resilience and to understand that okay, this is science is always. I understood that it is not about the excellence. It is about not an accident, but it is actually where your rigor, your curiosity, your collaboration, and your freedom to pursue the ideas. These are the, I think, words, or these are the terms which we remember, and that make us understand that you need to think beyond the boundaries and push your boundaries.
Yeah, that's nice. So
Absolutely.
yeah. So you trained after Max Planck. You also studied at UMass Medical School and later on at A-Star Singapore, right? What did
Yes.
each of these ecosystems teach you about impactful sciences or experimentation or the kind of data analysis you need to do?
So I would say that I was privileged. I was privileged to have very different scientific ecosystem, and each one of them taught me a lot of lessons, invaluable lessons about how can the science could impact. The UMass, when I moved from Germany, I always tell that European culture to American culture. American culture I found very competitive,
Oh, okay.
and you need to compete, you need to be the best. And I think I learned that how people work in collaborations.
Okay.
And there was a time when we are coming from a scientific background. We have weekly meeting from the clinicians. I remember every Wednesday 8 o'clock we are asked to come in the lab where all the clinicians are presenting the clinical work, clinical data. And that is where I think it opened my mind that we should not work in silos. We should not work. I am just an experimental guy, or a clinician is only a clinical guy. You need to intersect the science. You need to exchange the idea. You need to learn from each other. And this is where I started learning that when I came back India, I started questioning from the clinician that what is something which disturb you? What is something you want to do differently in the clinics, and how can I help you? This was the training where I would say the creativity, independence, bold thinking, and maintaining the rigor scientific standards. This is very important that when we think about the science, it has a rigor, it has deeper thought, and also I understood that how academia, industry, government agencies, they are all working in alignment. This is very important that you want to translate your discovery, science and technology. You want to commercialize your discoveries or your findings. And there's a partnership and an ecosystem which enables us. So I truly believe that the kind of ecosystem you go abroad, you see, and you want to come back and you want to develop similar kind of ecosystem. And perhaps a greatest takeaway for my understanding was the science is not measured solely by the publications or citations. I truly believe that it is more about reaching out to the patient, influencing the clinical practice, and create the solutions for the accessible and scale. You can do the things at a scale. Yeah.
Very nice, very nicely said. The way the collaborations happen, and as you said, scaling is quite important. During these years itself, you worked under globally respected mentors like Professor Carl Simon and Professor John Paul Thiery. What are some of the lessons that you took, and how was the guidance under them? And how did that shape your decision making skills?
Yeah, yeah. So I think I truly feel I'm blessed to work with these stalwarts, especially Professor John Paul Thiery, a very accomplished leader worldwide. He was a French scientist, and I always tell my journey that I worked and I got trained under a German scientist, an American scientist, and a French scientist. So you'll not find many such researchers who have been trained. Of course, in India, but from India to going to Germany and get trained and from a German, and he was again a very big stalwart, Thomas F. Mayer. I think he's one of the very known personality and authority in the field of infection biology from Germany. And then Carl Simon, who was a relatively young scientist, where I joined as a postdoc, and I think that was amazing journey because we had a conversations, hours of conversation about the science, and there was no boundaries because he started his lab, and I think I was his first postdoc. And from there I moved to Singapore, and Professor John Paul Thiery, I think I have utmost admiration that I always believe that I'm blessed to learn from him. His hard work, his the way he has a patience, and his I would say admiration and the way he used to work. So this is where I truly believe that these help me generating an understanding that how you can think about science, leadership, and innovation. I learned the value of thinking across discipline. And so extraordinary ability, to connect developmental biology, cancer biology, clinical relevance. You should not keep your boundaries just that, okay, what you are doing. You should interact with the people. You should interact, no matter you have to go and interact in the lab. You are interacting in the keyboard, coffee board, in the elevator. Yeah. And this is the scientific temperament and the time I felt that we are walking and we are discussing. Doesn't matter somebody is a physicist or a chemist or a biostatistician or a computational scientist. But perhaps I think the most profound lesson was the science is a marathon. It is not a sprint. People make mistake that, okay, I can achieve things very quickly. Great discoveries require patience, persistence, and resilience. This is, I think, it's a period that not every experiment works, not every manuscript is accepted, and not every hypothesis turned out to be correct. So what matters is to maintain the curiosity and integrity what you are doing. And I think, again, working in each of these countries, under each of these mentors, I really feel that as a scientist, as a mentor, and as somebody who is committed to translating innovation into the meaningful impact is very important. And I keep mentioning in every presentation of mine that working with John Paul Thiery changed my whole perspective about science and hard work. And the last thing I would say, the passion. I always tell that if you are passionate about your work, then only science require much more resilient and much more dedication, and you can only achieve if you have passion.
Nice. Professor John Paul Thiery.
Yes.
You had mentioned one unique experience you had, and which kind of completely disturbed your thinking process, and you tried to see how Dr. John Paul Thiery, Professor John Paul Thiery thinks, and you modulated yourself into a certain way of being scientifically different. So, what was that experience, and why? What was that?
So I tell you, I always tell, and I need to be very true, because when viewers listen this interview, they will know that how science and a scientist mind changes. I always believe that my journey become very serious when I moved to Singapore.
Okay.
I was doing science, but what was lacking is the touch. Even though I was in US or in Germany, but when I met somebody like Professor John Paul Thiery, I felt that with his age and his curiosity and his persistence about the work, and I remember that when I joined the lab, he gave me two very challenging areas to work. One is to discovery the biomarker for bladder cancer, and another one is liquid biopsy. And he told me that both these areas are very challenging. Do you think you would like to do the work? Because you have to compete the rest of the world, because there are much brighter people across the world, much brighter labs, and more funding. So that was something eye opener. But what has changed me about my perspective about the science, when I used to see him working hard, probably 18 hours, 20 hours, and one of the day and one of the evening I do remember, and I would like to mention that we were sitting on a manuscript correction, and I think it was 9:30 in the night when he told that okay, he's coming in the lab, and he was coming from NUS, National University of Singapore, where he was head of department of biochemistry, and he came and we sat with the group of people, and he started correcting the manuscript, and I think it was 11:30 something when we were like feeling very tired. But whenever I see his eyes and when I see his energy, I didn't find him that even a bit of tiredness. And I think we worked till 1:30 somewhere, and even we tried to know that okay, how we will go back to home, because we need to hire a cab to reach home. But all this is very small thing when you sit with the leaders and see the energy and you try to match the energy what he has. And I would say the another event when we were working on circulating tumor cells, and I just told, I called him and I told that I feel that the slides prove that I found a circulating tumor cells, and he immediately told that Prashant, I'm driving back. Let's stay, and he came back, and he used to spend hours under the microscope, and that's where I tell you the science is driven by curiosity and a purpose, and this changed my mind that no, I think I should be more focused because I am blessed to work with him. I am blessed to do something bigger and better, and these stories actually keep reminding me that I am answerable also to the science and the people who trust it.
Wow. You working under Professor John Paul Thiery, was that the changing point when you thought that right now it is important to think about patients and interact with them too, so that our discoveries are more profound?
So true. I think this was the first time I got a direct interaction, because the NUS and Singapore General Hospital, these are the two hospitals, and we started working on the breast cancer.
Okay.
And we were interacting with the clinician. We used to go, and I myself used to go and collect the blood sample during that time to these hospitals. That was the time I started believing that I'm dealing with the blood sample. The blood is coming from a patient, and I'm answerable to every drop of blood. When we were trying to culture those blood, to expand the circulating tumor cells, that started feeling more touched actually, that okay, these are the things which is not just a very instrumental, but these are the things which make me answerable, make me believe that I can do something which would impact the patient journey. And this there, when I moved to India, I started working with almost every hospital, you name it, in the oncology space, because I have been trained. I learned that what is the importance of your work which can contribute for the patient, and not only patient, but it's a journey which actually you involve with the patient journey also, with the family also, and you want to take an impact, you want to give life to the patient. So I think that was the turning point when I got exposed in Singapore to the hospitals, to the patient, and I carried it forward when I moved to India.
Wow, that's nice. So, doctor, a lot of researchers, they spend their entire careers publishing papers, but you chose a path focusing on translating discoveries into patient outcomes. Why did that come up, and what's the journey like?
So I would say that yes, we as a scientist, we as a researcher, we have been trained to do publications. And I'm nothing against publication, but the later point of time, I understood that I am not going to compete. Not going to race with the publications, that I have these many papers and how many papers others are published. Similarly, how much we discuss about the impact factor. And I think a couple of days back, uh, there was a posting from a very renowned clinician on Twitter about the publications and how much time it takes us to get a review. And we feel that it's the way AI is getting driven. Like AI is doing your review and they are asking the question where many of those questions are not making sense. So for me, when I moved India actually, and when I see the situation of cancer, when I was visiting hospitals and see that how many patients are suffering, that changed my mind that publication would anyway be the byproduct of your work or your discovery. But what is more important, that how can you translate your discovery from bench to bed and from bed to commercialize your discoveries? Because at the end of the day, no matter how many patent I will make, how many publications, but do I able to translate my discovery? Do I able to make an impact to generate a kit, even diagnostic kit, or a therapeutic vaccine or a therapeutic drugs? So I truly believe that this is the high time where the translational research is very important. And we all of us, it doesn't matter which domain people are working, either a physicist or a chemist or a biologist, but we definitely need to understand that publication should not be the core of research, but your discovery which meant to make an impact for the patient life.
Wow. Nice. So, doctor, you have worked on cancer biomarkers, genomics, circulating tumor cells, liquid biopsy technologies. Was there a moment when you realized your research could directly change clinical practice also?
Absolutely. I think biomarker, you heard this term very often, that how can you diagnose the cancer? There are multiple biomarkers are there. When I started working on the bladder cancer, we discovered the biomarker, and these biomarkers, I'm very proud to say that we made a US patent and Singapore patent. But it is not only the patent, but we able to commercialize our biomarker. There's a company in Singapore called BioCheetah. This company actually come up with the biomarker kit, and you can get over the counter. And this is coming from one of my discovery, converse your discovery from bench to bed. Similarly, the biomarker is one such area, and I think we worked, when I came back to India, I started working on circulating tumor cells, and this was with the Kidwai Cancer Hospital in Bangalore. Okay. And there we started collecting the blood sample, we started growing the CTCs, and we could correlate those findings with the patient outcome. So all such, either you talk about the genomics or you talk about the proteomics or the multiomics, the idea is that how eventually you can match, or how can you come up with something which could correlate with the patient outcome. And many such discovery, what I was mentioning and I'm mentioning right now, that these are the outcomes of the work which has been done in the lab in terms of different areas. Either you talk about the liquid biopsy, you talk about the biomarker discovery, you talk about the omics data interpretation, but it has a direct impact for the patient outcomes.
Wow. Wow. So your research and your identification has been still being used, the biomarkers itself, right? One of your contributions. So can you please explain what biomarkers actually do?
Yeah, so I'll tell you in a very simple way, and that what biomarker. Bio is in the term itself. It is going to talk about the biology, and a marker which can detect. Now, biomarker could be anything, and I keep giving the example that for the pregnancy test, it is the best biomarker world has seen. When you see the biomarker, when you find that the woman is pregnant, it means the biomarker level goes up thousands and thousands fold up. Okay. So HCG, we call human choriogonadotropic hormone. The level of HCG, fertilized egg secretes these hormones. Now you measure this. So when you measure it in the urine sample. And these are the biomarker which is non-invasive way, which give you accurately that, this is the finding. Similarly, in cancer, there are multiple such biomarker, what we discovered for the bladder cancer, and we actually compete with FDA-approved biomarker, which were complement factor H and NMP22. And our biomarker was the mix of 5 different protein-based biomarker, and we mixed and to get the high AUC, area under curve. The idea was that the cancer is very heterogeneous. So we started thinking that maybe one biomarker would fail to recognize every patient. So why cannot we come up with the 5 or 6 different biomarker and get the superior sensitivity and specificity. So any such biomarker, when you tell, the idea is that how can this biomarker test early, if it can tell us that okay, this disease is coming, disease is not coming. It could be even the liquid biopsy. And similarly, when we talk about the urine-based biomarker for the prostate cancer, PSA is one such biomarker which we use routinely, or that is the blood-based one. But there is a urine-based biomarker which is PCA3. So I think the saliva is another good example, or tears. So all these biofluid which we are using in the lab to identify and discover a biomarker which can detect the cancer early.
Understood. Wow. So this kind of innovations, I think you would have made a lot of them. How long does it take for you to start with an idea, process it, and for it to actually be in the clinical practice, or be it a drug, be it a system like biomarkers? How long does it take, doctor?
So I would say that I would reverse your question. I would say that the earlier, the way we used to think that okay, some ideas come and we process, but now I if anything's come, I go to clinicians. I told you that I think this is very important, that my work should impact the patient. Otherwise, for me, doing extraordinary work doesn't make any sense for me. So whenever some ideas comes, I truly go to the clinician and ask that does it make any sense? If I would suppose if I would come up with something and I ask you that okay, is it viable? Is it viable for clinician that you can use routinely in the clinics? And that is where all such work starts. It takes time. If you are talking time frame, there's always a phase. The any such biomarker discovery start with the discovery. And when you do discovery, it is a small cohort, probably 5-10 samples when you start discovery.
Okay.
When your discovery is done, when you identify, when you shortlist the biomarker, then you go back and do the discovery phase 1, discovery phase 2, then you go to validation and you start validating the small cohort, then you are going to validate in the larger cohort, and then then moving forward, you are going for the multi-centric clinical institutions where you want to validate very robust, because any such biomarker you discovery is very easy. You can discover something, but then the validation, it has to be validated in the larger cohort, and the validation is important because you want to identify a robust biomarker which has higher sensitivity and specificity. So it may take a 1 year time, probably minimal 1 year to 2 year or 3 year, depending on how quickly you will get the sample. But at least 1 year time for you to discover a biomarker and go ahead with making the clinical utility. It may take some more time.
Wow. So how does it feel, doctor? Like for when years of research, years of finally being in the lab and it coming to the reality, impacting the real patients, how does that feel?
I think it gives us a sense of responsibility.
Okay.
And humility, that if I would talk only about my achievements would only make sense if I would be able to make a single change, if I could make something I can contribute which can change the life of the patient. I truly believe that I hardly did anything and lots to do, because if I would do a small contribution which can change the landscape, or not even the landscape, but if I can change even the iota of cancer treatment or discovery, early treatment, screening, I would feel I'm blessed. And it demands lot of concentration, lot of dedication, lot of commitment and collaboration. And I truly believe that I have energy, and I think lots to do, and I would be looking forward many more collaboration, many more researchers and clinicians and brilliant ones, because I truly believe that in silos I would not able to achieve. The only way I would achieve that I work in hyper collaboration mode.
Wow, very nice. When did you come back to India, doctor? Sorry.
So this was very interesting. It was 2016, 16. Almost 10 years, 1 decade, I came back to India. And I really remember when I moved to Bangalore, The first thing I did, and you may not believe that first 5 year I gave my 100%. I was visiting every hospital, you name it,
Wow.
in the south part of India or north part of India, and trying to convince the clinician that this is the high time that let's work together. Because many of the samples which we do not realize, we just trash those samples, either blood or tissue sample. So I was just telling and convincing them that it's a biorepository. We need to generate the state-wise data. We need our Indian data, Asian data. And I think I'm lucky to convince some of the clinicians and work and generate work which is very much Indian-centric and not only work, but we published and very accomplished, journals who actually took our work and published it. But publication is one side of it, as I mentioned you from beginning. What we also try to do is that we would like to make the kit which could be routinely used in the clinic and it's affordable and accessible. I think this is the term we keep using it, but how many of us we are really planning and doing our hard work to make it affordable? Because at the end of the day, when you discover and you come up with a very smart kit, but if the patient cannot afford or if cannot pay for any such test, then I am not successful, because it should be reach out to the mass population.
Very true. So that is what is your phrase from bench to bedside actually means. You established a successful international career, came back to India, but when you came back to India, you started off with Ramanujan Fellowship. Why was that important, doctor? What was it about?
So I think I must thank DST, Department of Science and Technology, and DST allows to bring the brilliant minds from abroad to India. And I applied for DST fellowship. It covers your salary, and also it start and gives you the seeding funding, that you can start the lab, you can pursue, and you can work.
Wow.
And I think when I got the DST funding, I was very confident that I have some funding. I have my salary, because I am not dependent to get salaries. And they fund for 5 years. I think this is enough time. And I think before even completion of 5 years, I got independent. I got multiple other opportunity and positions. So I would say that it was a hard work, dedication, and learn. I was very curious when I came to India. Suddenly my landscape changed when I was roaming things in and meeting people, their different mindset. When I came India, I had a struggle. I would not say that it was a catwalk and I was really getting everything on platter. No, it was not. Struggling, going to the institution, meeting the clinician. But I feel I'm blessed to meet some of the stalwarts, some of the amazing, extraordinary clinician in India who supported very strongly. And from me, from academia, I moved to industry, because I felt that you want to pursue bigger dream, and you do not want to be limited with a small amount of money and changing your dream and becoming assistant to a professor, or you are just thriving for getting grant. I felt that industry where I can pursue my dream, I can do something beyond my thought process, and this is where it shaped me when I actually moved India, getting the Ramanujan Fellowship.
So, what kind of cancer care realities in India surprised you? And what were those discoveries?
Yeah, that's a very interesting question. That what is something surprised me, and I think if you talk about the cancer, especially the hospital abroad, and you find that things are very structured. When I came India, I found that okay, we need to improve a lot, and of course I would not blame or claim a hospital or the system, because our population, we are now almost 1.5 billion people. So to cater the need of these many people, every hospital, especially government hospital, it was not easy. And many of the top OPDs, if you go and see the clinicians, they are loaded with the patients. But at the same time, I realize that many of the things are not, they cannot afford it, because the affordability is a big issue. And how people can afford the test, how can they afford even the best of the best discovery what have been made. So I think when in India I moved, I was going in many big hospitals, starting from AIMS or PGI Chandigarh or Kidwai Cancer Hospital, and I found that there's a lot of challenge. And the challenge is that how can you structure things, how can you place things, and how can the patient would get the utmost attention. I truly believe that over the time things are changing.
Okay.
I think the clinicians are putting a lot of effort. Research are going on in most of the people are involved. They want to do the collaborations, and I think this is what is needed. That we would come outspoken and share the data, talk to each other, because that's very important that we do not talk each other. And within a hospital also you find that people are not talking, the clinicians are not talking, the researchers are not talking. So that is very important that
They
this is not about
collaborate and
collaborate. They collaborate, and this is not about only my journey. It's a patient journey, and we can only be successful when we have the collective effort. Doesn't matter patient part, scientist part, or the clinician part.
You said India has a lot of patients, and every doctor is overloaded with the kind of patients that they get, be it OPD, IPD, anywhere, right? What do you
Yes.
think is the biggest advantage for India today in precision medicine and cancer care innovations?
Yeah, I think we are in a very advantageous situation. And many times I tell you a very good example that in Singapore, we were struggling to get the samples.
Oh, okay.
A small country with high and high level infrastructure, but and the patients are not there. We have extraordinary learning. Imagine the diversity. Every state is a country.
Correct.
So now you have such a diverse population, such a diverse landscape, and have the energy to actually dissect and generate your own data, because it's very important.
Yes.
So far, we are relying the data coming from the Western world, and this is the time we need our own genomic data. We need to understand how differently we are. So I truly we are in a very advantageous situation. We have brilliant doctors. Trust me, that we are the country where the doctors are amazing. The scientists and many scientists coming back to India from abroad, getting trained. And when the youngster asked me that, would I suggest them to go abroad? I tell you now, maybe not. When we were going, yes, because the India was not matching up to the technology. But we are very study. We are doing as good as any other Western world can do. We have all the technology present here and brilliant mind. So I truly believe that we are in a very advantageous situation where we can guide the world, and we can set a benchmark that how the precision medicine could be used, because we have the diversity of the patient, we have the treatment opportunity, and we need to learn from each other.
So, doctor, cervical cancer is one of the innovations that we as a country have done a lot. And I think you also are part of that team which did a lot of study in cervical cancer, and you have come up with innovations that can make it, that has made a difference in for the patients out there. Can you please explain what your innovations have been? What has been the journey?
So I think this was a very exciting journey. 7 years back, I think I got a collaboration with Dr. Richa Chauhan from Mahavir Cancer Sansthan. And I always praise her because I think we need many such amazing clinicians who are self-motivated. And I think she kept telling me, Prashant, what is something next we can do? But the cervical cancer, it is still a big problem. And especially country like India and Africa, where we do not understand the very basics of the basic. How you can maintain the hygiene. We do not have understanding about the virus. And I think the question was raised by the clinician that the patient who are treated with the concurrent chemoradiation, it's the first line of therapy where patient stopped responding, and the data suggests almost 40 to 50% of the patients stopped responding to the concurrent chemoradiation. And that is where she raised that can we identify a prognostic biomarker or a predictive biomarker? And can we also do the genomic landscape of cervical cancer, because there's no data worldwide at the whole genome sequencing level. So we did it successfully. We discovered the biomarker STX3, and this is patent. We got the Indian patent, and now we are working to commercialize this biomarker. We're trying to develop a kit, and the kit would be very simple. It could be used any part of India, even in a very remote place, because we are not trying to develop a very hi-fi science or putting a instrumentation, but it could be either very simple ELISA-based test where you could just predict the biomarker based finding that if the patient is going to respond to the therapy. Another finding what we did is that we contributed for whole genome sequencing of cervical cancer, and there's no data even at TCGA. So we are first globally discovered, and we have identified beautiful data. We identify a target for the drug, for the patient who are not responding. What are the kinase which are getting hyper phosphorylated, and can we develop a drug against that? So
Yeah.
I would not go much detail because we are working on this to develop the drug, but the goal is that we would like to bring the innovation from the Indian data, from the Indian discovery, and can we change the whole practice of the cervical cancer, where new drugs could be also, used for the targeting those particular kinase molecule in cervical cancer.
Wow, truly proud of that. I mean, something made in India and developed with the genome data of India. I would advance congratulate you and also wish you all the very best.
Thank you.
Okay. Now, what still needs to be changed in India, even after so much of innovation, so much happening? What do you think needs to still be changed in India?
I truly believe that we need to be very focused. We need to be hyper collaborative way. I keep telling that we go and talk about many such forum. We talk about how industry and academia can work,
Yeah.
but we only talk. After the meeting, we forget. We do not pursue. And similarly, industry comes because we need to scale. If my discovery is found in the lab, if I'm not able to communicate and translate my discovery from bench to bed, it's not going to work. It's not going to reach to the people
Masses.
or the patient.
Yeah.
Exactly. So I think that there are brilliant mind. I told you there are multiple beautiful and good institutions across the country. But what I feel is missing is that communications. We need to be more communicative. We need to be more transparent. We need to work for a purpose. And I always tell that the science should be purpose driven. It's not about what I think. Prashant Kumar could be one such individual, but we need many thousands and thousands such individuals who are coming and putting collective effort in science for a purpose. And the purpose is that how can we do something for the cancer patient? The way the numbers of cases are increasing, we predict that there will be surge more than 70-75% surge by 2050. So you can imagine that how can we do such training? How can we train people that what need to be used, what not to be used, what need to be eat, what not to be eat? We need to make awareness about the cancer. Early screening is something which is missing, and most of the time in India, the cancer present at the very latest stage, which is not the case in Western world, and this has to be changed in India. So I truly believe that more collaboration at hospital and research labs. Every hospital should have research going on and publishing, not only publishing, but also find a ecosystem where agencies should work together. It is private government partnership. The agencies means the regulatory body. They should be very open. They should come and give more opportunity, because this is the time when we talk about the cutting edge technology. I think we should accept that many such technology need hand holding, and many experts should come and guide and make the path which would actually pursue us to take it many of the discovery viable.
Precision medicine, doctor. We have been hearing this term, but for somebody who is not into the science background like me, what exactly does precision medicine mean?
Yeah, we have been hearing this word, and the way we were treating cancer, it was for one disease. Like there was no specific, or I would say that very generic way we were treating. We are using chemotherapy, and chemotherapeutic drugs is used for, still we use for 70% of the cancer treatment. But precision medicine term came when we understood that we have to be very precise. We need to understand genomics, genomics of cancer. 2% or 3% coming with the same cancer, same disease, same symptom, may have a different mutation. So you cannot pick a cancer, somebody presenting with the lung cancer or the breast cancer, and you treat that, okay, this is the line of treatment. And this is where the precision medicine came, that we call it N is equal to 1. So every patient has to be treated individually. We need to spend time, we need to learn their history, that how it is different from other person.
Okay.
A person, how the person got exposed to the cancer. Do they have the genomic coming from the hereditary, or it is somatic mutations? But until unless we understand the pathobiology of the disease, this is very important. That the precision medicine allows us that there is no one fit drug or one drug fit for all cancer. There are drugs where we are talking about the tumor agnostic also, but it also comes with the studies, with the thousands and thousands of data which suggests that if particular mutation is there, this drug may work. But we talk about the mutation, and I'm talking about the variants. But there are multiple. One mutation may have 30, 40 variants, and each variant is going to act. So I think precision medicine is precise. The term itself called precision. So we need to be very precise to go deeper, to understand the genomics, to understand the mutation, to understand the alterations. And this is not at the level of genome, but it is level of the transcriptome, it is level of the proteome, it is level of metabolome or methylomics. And we are in the era when we call multi-omics.
That was an amazing explanation, doctor. So, in simpler terms, as doctors and clinicians and scientists like yourself, you identify what exactly is the cancer or where exactly is the cancer breeding, what kind of mutation and variance is happening, and what kind of cell breeding is happening, and then for each of the variants you kind of define what the treatment could be and what the drugs could be. That is precision medicine.
Absolutely, absolutely.
Okay. So your career has spanned across academia, healthcare, startups, foundations, and industries, and you have been exposed to multidisciplinary departments or multidisciplinary things, right? What shaped your approach towards innovation?
So I truly say that the science and the scientific endeavors need lot of exposure, and again I'm blessed to have such kind of exposure, starting from multiple top institutions, from Indian Institute of Science to Max Planck, UMass, Singapore, coming back to India, again worked with Institute of Bioinformatics, then worked with Vidya Bihar as a dean, and then industry where I worked as a chief scientific officer of Datar Cancer Genetics, and previously I was working in Arkinos Healthcare. I also founded a Liquid Biopsy Foundation of India together with my colleague, I call her the person who showed me path as a clinician. She was a leader, Sevanti Limay. We together founded Liquid Biopsy Foundation of India. A startup also, have been working with multiple new startup, want to do work in collaborations. All this came for a reason that I realized that there's no any one way you can succeed, you can achieve. And we as individual, the time came when we should be open for everything what comes in your plate.
Yes.
So people have told that why you need to do only, there are people who works whole life in one particular science or one particular area. And if you see the strategy or the approach where I started my science, when I started with the infection biology, then I came to cancer biology, again cancer biology, I started working on almost 4, 5, 6, 7 cancer, where you'll meet many people who work whole life in one particular cancer or cancer subtype. I truly believe that I should not limit myself with such a type of cancer or subtype of any particular disease, but I was always open. I need to see the 360-degree angle, a holistic, because that will give much more idea about the disease biology, to learn from each other, and you learn science. When we call that the era where the multi-omics is something which we practice and which we will practice, it could only come when you have the learning of genomics, then transcriptomics, then proteomics or metabolomics or artificial intelligence. So I truly believe that all this journey where I have been, part of many such institutions, part of many such leaders or stalwarts who trained me, that give me a value to understand the importance of multidisciplinary science and how can I utilize my science, my learning, my expertise, so that holistically I can grow and I can have a far better and deeper picture about the science.
Got it. You said Datar Center, like cancer genetics and Datar cancer biologist. You are currently working there. You have helped advance the biopsy, precision medicine, and cancer vaccination. cancer vaccination. What excites you most about all the work that you have carried out at Datar?
So I think cancer vaccine is one of the area I have been dreaming to work for a long time. And when I moved to Datar Cancer Genetics, one of the projects which was discussed about to develop the therapeutics vaccine. And when we call about vaccine, it is very important to understand that there are two kind of vaccine. One is preventive, and right now for cervical cancer, when we talk about the vaccine, it is HPV driven. The vaccine we give to the young ones. Actually, these are the vaccine which allow not the or which act against the HPV virus. This is where any such vaccine which we develop, and very good example is COVID. COVID is one such example where you could see that we were highly dependent on the vaccine. Either it was developed for the mRNA-based vaccine or protein-based vaccine. But the idea is that how can you prevent the disease? When I am talking about the cancer vaccine, I'm talking about the therapeutic one. And the therapeutic vaccines, when we tell, this is against the progress or the progression of the disease. That the disease, already the person has cancer, and how can you re-regulate, re-stimulate the immune system of the patient, where the immune cells would be highly active and acting and killing the tumor cells. So this is where I am very excited at the very early stage, and looking forward that we will get the clarity very soon and an approval to go for the phase 1 or phase 2 clinical trial in India.
Wow. So I think this could be a revolution, right? So, doctor, you also were mentioning about liquid biopsy.
Yeah.
It is I understand from your words that it's kind of a game changer in oncology. What exactly is it? Can you please help us understand?
In a very simple term, when we talk about the biopsy, it gives you a clear indication that you are dealing with the biopsy, means tissue sample which has to go through the surgery. And biopsy, whenever you do biopsy, it's not a very favorable feeling, because there is a surgery involved.
Yeah.
And this has to go, and if you want a tissue multiple times, you have to just imagine that the patient has to go multiple surgery. Liquid biopsy is a revolution, I would say, and I'm very, very much into the liquid biopsy. I have been working almost 1 decade now in the liquid biopsy field.
Okay.
And when we call liquid biopsy, it is in a very simple way that you would get similar kind of information what you would rather get from the tissue biopsy. And liquid is in a way that it is minimal invasive or non-invasive way. If you would get the kind of pathobiology information, disease information, and the biofluids could be either it's a blood sample, urine sample, saliva sample. So depending on what cancer type we are talking, if we are talking about the bladder cancer or prostate cancer, urine makes sense. If we are talking about the head and neck cancer, saliva makes sense. And blood is something amazingly important, because the blood where the tumor cells shed, and there identify the tumor got in necrosis or apoptosis. So what you would identify, that circulating tumor DNA. And now tons of data, tons of information validated that the liquid biopsy is evolved. It actually transformed the clinical practice, and you could use liquid biopsy for not only early diagnosis but monitoring of the disease. You can identify the resistance mechanism, and also you can actually estimate the relapse of the disease. So I would say the amazing future of the liquid biopsy, where we can detect the MRD we call minimal residual disease after surgery or treatment, to identify relapse. And I would say that we can have longitudinal samples. You can ask a patient multiple time blood sample, but you cannot ask a patient multiple time tissue sample. So this actually changed the whole dynamic, the monitoring of cancer dynamics, managing the chronic disease and through the regular blood test. And I think it moved from waiting for tumor or the radiological. We are going in an era where the liquid biopsy would change the whole perspective of the disease management.
And all this has been developed in India by yourself and your team.
So this have been developed across the world. There are different niche area. I have been working much more intense in circulating tumor cells. I started this work from Singapore, and this work was very successfully we established. I moved to India, and Datar has developed multiple such tests, and one such test where they do based on the circulating tumor cells, where they have developed a method to culture these tumor cells in the microwell plate. And these microwell plate where the tumor cells are there, we actually exposed with the different drug combination, and you can maneuver the different drug combination to identify which drug would be selectively working for the patient. And this is where we call precision medicine, because this is giving you a very deep information about the drug susceptibility testing, using the drug susceptibility testing, that what drug would be effective for the patient. So I would say that liquid biopsy is the future, and Datar Cancer Genetics definitely contributed immensely for the development of liquid biopsy test in India.
Wow, that sounds really amazing, and I'm looking forward for that, for the next kind of innovations that you are bringing out in liquid biopsy. So, how do genomics, artificial intelligence, multi-omics, and digital health come together to shape the future of cancer diagnosis and treatment, doctor?
So I think this is very extensive topic,
Okay.
where we are trying to accumulate the data from the genomics, the liquid biopsy, artificial intelligence, digital health. I truly believe that nowhere we would grow just using one arm, just only doing genomics. As I'm mentioning you, the time came where the cancer is a very complex disease. And every time we dissect cancer, we understand that we know very minimal. And still with the era when we talk about the multi-omics and AI, our understanding of cancer is not more than 30%.
Oh,
So you can imagine that tip of iceberg, and still 70% need to be unrevealed.
wow.
So I truly believe that when we are talking about the complexity, we would need the data at every stage. And this is where I think the future is, where multi-omics would be used, where we would learn the cancer biology, not only by doing genomics, not only learning about the genomic alterations, but also the gene expressions, also the protein expression, also the metabolites present in the cancer environment or the tumor microenvironment, understanding the immunology, how the cell behave, or the cellular behavior of the cancer. So I think there's a extensive way where artificial intelligence, when we talk, the idea of artificial intelligence is that AI can process the data quite extensively and rapidly, which human not. And this is where we have to integrate the multi-omics, the liquid biopsy information. I told you that many times people can answer me those areas which we may miss out with the tissue biopsy also. So I think integrating all this omics-based data, liquid biopsy, and AI-based data, we are looking to develop an algorithm, we are looking to develop a program or a risk score pattern which could actually guide us to give a very authenticated way to diagnose the which can handhold us for therapeutic decision making, and that could be only possible that when we are integrating the data at every stage.
Nice, amazing. Doctor, when you say that we have a lot of patients in India who can help you gather the data required, right? It's directly indicating that there is an increase in the number of cancer patients in India. Like, as a scientist, you would have studied the causes also, right? Why is it happening?
I think this has been asked to me very often whenever I go, especially in the meetings and when the people interact. As I told you previously, that the cancer will grow and it's growing. And each of us know somebody in the family or friend where they're suffering from cancer.
Yes.
This scenario has changed in 2 decades. 2 decades back, it was not that often. Cancer was called a rare disease, and now it is called prevalent disease, or most prevalent disease. And I think a lot of information we collected over the time, and our understanding improved. Maybe the cancer cases were earlier also, because now we understood the detection. We have much more method or the methodology which could be help to detect cancer pretty early, or we can diagnose the cancer. But there's something which we understood, that the use of pesticides, as you know that we are using lots of pesticide. or even when we talk about the milk product, these all milk which we are intaking is full of hormones. So if you see the pollution, I think recently there was an article came, pollution cause cancer. You are working in the printing industry, or somebody working with all such industry where they are exposed to chemicals, so we are much more exposed to have the cancer. So there are multiple reasons, the kind of lifestyle we are living. We do not want to move. We are all dependent on computer, and the fingers tip, we want everything to happen. So I think a lot of things coming because of our behavior issue, the environmental issue where we are living the life, and the lung cancer cases are increasing. Smoking is one such big reason to cause cancer. It can create at least 14 different kind of cancer. So it's chewing tobaccos. So, chewing tobacco, where you recall that the earlier people used to chew tobacco and keep tobaccos for hours and hours
In the mouth.
below in the mouth, and now they are they are on the risk of getting oral cancer. So depending on what kind of cancer we are talking, but there are multiple ways we could actually minimize, at least, other than something coming genetically which we cannot. So the only way is that you and all of us need to go for early screening, and as routinely we can screen the cancer. But the number of cases are increasing because of the adulteration in the food, because of the environment we are living, because of the lifestyle we have adopted. All changed.
I mean, there are multi-facets to the cancer itself. That's what I understood now. If India has to become a global leader in precision medicine and cancer innovations, what could happen or what should happen over the next decade, Doctor?
I truly believe that India could guide the world. We have an extraordinary set of people, the scientists. We have people. We have top clinicians. We have the population. We have diversity where we can learn a lot. I think what is needed is the collaborations. The infrastructure has been created. We need to use the infrastructure. We need to share the infrastructure, and we need to go in the early detection mode. We need to adopt this in our routine practice, that India could invade the cancer, and that could only invade if we start picking the cancer early. We need to also invest our time into the research R&D. I keep telling that India should be involved in the R&D activity, and academic, clinical, and industrial partnership. I think this is much more required. This is the time that we need to go for translational ecosystem, where discovery need to move from lab to or from bench to bed. Clinical trials, I think India need to spearhead. We are not doing the phase 1, phase 2 trials. So this is the high time that India should lead the clinical trials, and also clinical trials with the regulatory agility. I always find that we are bonded with the regulatory processes. So that need to be pursued and thought of it. And at the end I would say that the success should not measured by number of publications or the patent. It should be innovation. What innovation you develop, and this is required for India to improve the lives of the patient. When we working as a researcher, working as a clinician, the goal is not only the publication or the patent or unicorns, but it should be that how many patients we are saving the life, how many innovations we are doing.
Very true. So, Doctor, you have been traveling across the world. I think a couple of days back also you were in Boston. So,
Yes.
and you have to handle, you said you have your own startup, and you're also heading Datar, where you head the scientist department in oncology. And you have been writing publications, although you say that it is not important. I mean, it should not be the measure of success, but still publications are important. And you also are reaching out to the patients and clinicians as part of your research and as part of your testing. So how do you manage your day, and how does your day look like?
Wow. Wow. This is something very exciting question. See, I have very unorthodox way I work. I tell
Okay.
you, there's no as such routine. There was a time I remember when I was working in Karkinos Healthcare, and I had. You can always message me and I will respond, no matter it is 2 o'clock, 4 o'clock, and people think that I'm sleeping with laptop. So, and this is not thoughtfully doing with the laptop sleeping. It was that the curiosity and the responsibility when people are working with you, and if they ask you any question and you would able to answer it, even it is in the midnight. And this I learned again from my mentor. I always remember John Paul Thiery, 2 a.m. also, if I write him, he responds me back. So this is something I learned from him. But the way your life and the way you planned your day, I think science is all about passion. Trust me.
So there's
It
no
could
planning.
not be, yeah, it could not be this fat salary and then I have to work. Or if I'm getting very small salary and I am not going to work because my salary is less. It is about the passion which drives you every day.
Okay.
When you see patients are dying day to day life, you feel that okay, you cannot do. And many of the days when I find the patients dying, I hate myself. I feel that I have not done anything fruitful. I have not done to save the people. So for me, doing and roaming around the world and talking that I am very accomplished researcher and scientist doesn't make any sense to me. So I truly believe that the routine, I would say that I am 24/7 available. If there is a reason, I try to do. I have the group whom I interact. We have, of course, we have a mission. The mission is that, okay, we would like to develop something Indianized, which could be affordable and accessible. We do publish, as I told you, that publication is not, and it should not be quantified that you are succeed or you are successful. But sometimes publication is required because your paper could be cited across the world, across the globe. People can read your paper publications, and I, at the same time, I urge everybody that publish the work, let the world see, let the world challenge your work. Because if I am sitting in my silos, I'm sitting in my own world and think that I am doing everything correct, I may not.
Yeah.
And this should be actually challenged. So the beauty of science, I always tell, the beauty of science is that it has to be challenged, and it is challenged.
Yeah.
When you write a manuscript, peer reviewed, they are experts, read your manuscript, they question you, you answer it, you rebuttal it. So I truly believe that my time goes from day to night. Whenever I am very excited, I just go and forget what is happening in my surrounding. If not, then I am just like you or any other person who is living the life, and I try to understand and learn day to day.
Amazing. So that indicates that you are dedicated to science and oncology and helping improvise lives. That's excellent, doctor. So looking back at your journey from a curious student at IISc to the global cancer researcher and scientist now, what has been the most rewarding, and what message do you intend to give to the future or the next generation of scientists who would want to enter the space?
I would say rewarding is that I have been chosen. I always tell that I'm blessed. I keep telling every time I'm blessed because it's a 1.4, 1.5 million billion population. So competing with these big number of people, and there are many more intelligent people, probably maybe more intelligent than me, but I have been picked, I have been given the platform. So I thank God every day that God has chosen me. Now it is my responsibility. At the same time, if I have been chosen, then it's my responsibility to do a honest work. And this is where I truly believe that the opportunity I got, the people I meet, I try to learn. I try to inculcate good things from them. I may mistake. I can make mistakes, and I always tell that a human are bound to make mistake. We cannot tell that we are ideal. Nobody is ideal, but we need to learn. Today we need to think that okay, how can we improve? And improvement could be anything: your scientific thought, your scientific process, scientific mining. And I think this is where I would say that the future is bright for all of us when we dream big. So for the youngster, I would say that dream big and read. Reading is very important. Read widely, not only science. You can read history, you can read philosophy, you can read biology. Because I truly believe that the knowledge grows it should not be unilateral, but it should be multilateral, and it shape your curiosity across the discipline. This is very important that when we read, we will a reader, we learn many things. It doesn't matter you read a novel, you watch some documentary, or even a movie. But the idea is that each of these process you try to curate something which you think will be used going forward. So I would suggest at the end that the youngsters should have the practice to read, and as much as they read, they would see that the learning will only make them grow.
Yeah.
What they were 2 years back or 5 years back, and coming forward, they would see that there is an improvement, there is application of the thought process which they can implement whatever science they are doing in whichever part of the world.
Wow, that's truly insightful and an impactful advice that you have given, Doctor. Thank you so much for all the insights you have provided, right from you choosing science because of the Dolly the sheep and the perpetual clone that was made, to the point of you traveling to Singapore under training under Professor John Paul Thiery, to the way you explained the liquid biopsy, the different kinds of innovations that you have made and contributed to the world. It's something to take away, and I genuinely had a great time talking to you, understanding the nuances of the scientific processes and policies and systems that you have undertaken and taken forward also, and all the innovations that are in the world out there because of your efforts. Truly salute you for all of that, Doctor. Thank you so much for your time, and really appreciate your insights and the impactful advice that you provided. Thank you so much.
Thank you. I really appreciate your time too, and I think the conversation will help many youngsters to understand the path, the journey, and they would do far better job than what I have done. So looking forward. Thank you very much.
Yeah.
Thank you.